Okay, so now we've established the question that we want to ask. Does the length of mollusk shell differ among populations? And we know based on the types of data we're using and based on the number of independent variables we have that we want to run a one-way ANOVA. Again, it's called one-way because we're using one independent variable. So let's go through how to actually run this test in SAS Jump and also how to state our results of this test. So I'm going to clear this out and I'm going to go to my Excel sheet. As you can see here, I have this mollusk data. I have the population of each individual as well as the length of the shell for each individual. So the first thing I need to do is save this document. And again, whenever we save in Excel, we want to be sure mollusk data. We want to be sure to save it as an XLS file because that's the only file type we can transfer into SAS Jump. So I've saved this and I can just close that out. Now I want to go to SAS Jump and again I want to open a new data table. Go down and find my mollusk data and open it. And I'm just going to say import. All right, and now I have all of my data in my SAS Jump file just as I needed it. The first thing that I want to do is to check that my columns are appropriately labeled, the data type is appropriate. So we know this population is a nominal data type. So we go over to our left-hand side and we see that yes, it's properly labeled as a nominal data type. This red histogram, the red bar graph, shows us that that data is nominal. If for some reason, let's say it was a green histogram, well, this is not the right kind of data. That's ordinal. So all you would do is click on that little green bar graph and change it to nominal. All right, our shell length is a continuous data type. It can take on any value between a min and a max number. And we see that it is appropriately labeled in SAS Jump by the blue triangle. Okay, so let's talk about how to analyze this data. The first thing we want to do is go to our Analysis tab, and then we want to go down to the Fit Model platform. Now you can do this analysis in the Fit Y by X platform, but in the next series of videos we're going to be talking about a two-way ANOVA, and in order to do that you have to use Fit Model. So we're just going to use Fit Model for the one-way ANOVA as well. So we click on that, and we see our box that we saw in a prior video. Over here we have our three columns, observation, population, and shell length. We know that shell length is going to be our dependent variable, our Y variable. So we can either drag that over into that box, or if you prefer, you can click on the shell length on the left-hand side and then click on the Y, and it'll give you the same effect. Where we see construct model effects, this is where we're going to put our independent variable, which is population. So I'll just drag that down. And we want to be sure that this says effect leverage. Then we'll click run. All right, so here are the results you get from the SAS output. Now, we talked earlier about statistics and how we use p-values to determine how we should interpret those statistics. And we said that if a p-value is less than 0.05, we consider there to be significance. In other words, we say there is a difference among means. So how do you find that information in this big window of data? Well, what you want to do is go down to where it says effects tests. And sometimes you'll see effects tests, but there will be no window underneath it. So all you need to do is click on this little arrow to the left of it, and it'll drop a menu down. So what we see here is our F ratio, which is our test statistic, and then importantly, we see prob greater than F, and what this is our p-value. So let's make a note of that, just so it's easy to remember. The term prob greater than F, when we're talking about SAS Jump, equals our p-value. Okay? All right. So we know that we have a p-value less than 0.0001. What does that tell us? Well, it tells us that our means are different because the p-value is less than 0.05. So how would we state this in a results statement? Well, let me push this over here a little bit and let's talk about this. So whenever you have your results statement in a paper or in a presentation, it's not enough to say there was a significant difference among means. Yes, that's what we learned from our statistical test, but that's not showing any of the biology behind the work we did. So instead, we need to go over to this area, the X3 population, which is the independent variable we used. And underneath this, we can see our means, also called least square means. And we can see that Petersburg was quite a bit larger than all of the other populations, the shell size. But it's also going to be very important for us to determine, yes, we definitely saw bigger shell sizes at Petersburg. So basically, whenever you see this p-value, all that's telling you is that there was a difference somewhere among the means. It doesn't tell you which means were different. So how do we know exactly which means are different? Well, we can do an a posteriori test. Now, we're only going to be doing this type of test for a one-way ANOVA, but your histomentors can help you with this if you'd like to do this test for other analyses we do. So the test we're going to do is called Tukey HSD, or other words, Tukey's honestly significant difference. And this tells us which one of these four means are different from the other. So in order to do that, we go to this red arrow to the left of population. And just so you know, it will not always say population. For example, if you were looking at gender, this would say gender rather than population. It depends on what your independent variable is. But we'll click that red arrow and we'll go down to LS means Tukey HSD. And this is the test we'll use. All right, and that brings down a whole nother list of information. So just for simplicity, let's skip over this information and go straight to the bottom. If you see here on your left, you have each one of your populations: Petersburg, Portland, Tillamook, and Newport. You have the mean of each one of those individual populations, but in the middle you have a series of letters. And if you read below that, levels not connected by the same letter are significantly different. So what does that tell us? Well, we see that Portland, Tillamook, and Newport are all connected by the same letter, the letter B. So we can say that none of those populations are different from the other ones. And yes, you see different numbers for the means, but in order for them to truly be different from one another, they would have to have different letters, and we don't see that. So there's no difference among these three populations. However, there is a difference in the Petersburg population because it has the letter A. So how would we state these results in our results section of a paper or in a presentation? Well, like I said, we always want to include biological significance. So a typical results statement that I would use, so results statement would be mollusk shell lengths differ among populations. So mollusk shell lengths differ among populations, with shell size being largest. So with shell size being largest at the Petersburg population. Now, one thing you always want to include whenever you're writing a results statement are your F statistics and your p-value. In order to do that, at the end of your sentence, you just put a parenthesis and you put F equals, and let's see what F actually equaled. So right here is the F statistic. F equaled 15.0511. So 15.0511, comma, and P was less than, as you can see over here, 0.0001, close parentheses. And we do that just so people know what our results are and they're able to trust our results statement, because we're saying, based on these analyses, based on this test statistic and this p-value, we know that mollusk shells sizes do differ. Okay? All right, so one thing that I think it's really important for you to understand is a results statement is completely different than a conclusion or a discussion statement. So in a results statement, you're simply stating the facts. You're not trying to account for why that conclusion may have come about. You're simply saying this is what happened. So a discussion or a conclusion statement is different. In those discussion and conclusion statements, maybe you need to think about why this larger shell size may have occurred at that population. So discussions and conclusions try to explain the results. They do not restate the results. So maybe I'm thinking about reasons that we could have found this result, that mollusk shell lengths were largest at the Petersburg population. And maybe there's more calcium in the Petersburg area. So I could say something like this for a conclusion or discussion statement. I can say, for conclusion or discussion statement, the largest shell size at the Petersburg population, so the large shell size at Petersburg population could be attributed to abundant calcium in the water. So do you see the difference in the results statement and the conclusion or discussion statement? The results simply state factually what we found from our analyses. Mollusk shell lengths differ among populations, with size being the largest at the Petersburg population. But then in our conclusion or in our discussion, we talk about why these shells may have been larger at the Petersburg population. Okay, so the next video I'm going to do is going to explain how to graph the results of this one-way ANOVA in Excel. So that will be up next. Thank you.